Ladies and gentlemen, what do you think? Can we do this in one take? Open up to 176 in your notebook. Today is Friday. Today we're going to take a look at leaf physiology, root physiology, and stem physiology, not necessarily in that order. And physiology means the anatomy of a object or a living thing and how it works. So we're going to look at how those work today. We're also going to do homeostasis, and along the way, I'll point out relevant vocab. You can use this to work on your answers to these. And as you're doing this, Mr. Diaz or I will pull you out for your interactive oral assessment. This is just a formative. You're practicing. We'll give you some feedback, and it'll be a good day. So when you get pulled out for your IOA, remember, answer the question, give me details, connect it back to the question, and make sure you include your vocab. All right, folks, here we go. First things first. Let's start with root physiology. Everybody turn back to 181. I've got some more things to teach you about this that we decided weren't worth all of our time on Thursday. So we've got our root here, and you may notice that there's sort of three distinct sections to this root. We've got these larger cells up here, we've got these smaller cells down here, and then we've got this sort of lump of cells down here at the bottom. So here's what's going on. Right here at the top, you can see just a small oval of cells. This is the vein of the leaf developing. That's what's going on right there, right? Or excuse me, the vein of the root developing. So let's add that in. This is the vein developing. All right. And then you've got this section where the cells are clearly mature. All right. They've sort of stretched out. The nucleus is very visible. But then down here, you have these much smaller cells. And what's happening here is that these are basically baby cells. They're still maturing. All cells start in this area right here, and you can see there's a dense little area with lots and lots of nuclei. That's because this is the meristem. This is where baby cells are born, okay? And this is where mitosis is actively happening to produce those new cells. So let's label this meristem. Okay. And that's where the cells divide to grow the root. So when a root gets longer, it gets longer because the cells are dividing here and it keeps growing farther and farther and farther into soil. These cells are never going to go anywhere. We're just adding new cells to the end. Now, obviously, in the soil, there's a lot of rocks and dirt and very abrasive materials. So this chunk of cells at the end is basically a helmet for the root as it grows. These are our new baby cells, and they're really tender. But these cells out here form what's called the root cap. Oops, I forgot to underline meristem. So that's our root cap, and it protects the growing meristem cells. And so that's how the root sort of pushes its way through the soils. This root cap works like a helmet, and the meristem behind it just keeps dividing and dividing and dividing, growing more and more and more cells, lengthening that root. Do you like having like a weird little Miss Ire in the corner? Honestly, kind of freaking me out. All right. So right here, we've got these little baby cells. They're really tiny little boxes of cells. Right? That's what's going on here. And then if you look up a little bit, you'll see that the cells kind of stretch out and they start to lengthen. That stretching process is also part of the growth of a root or even a stem on the other end of the cell, because this is the root meristem, but there's also what we call an apical meristem at the very top of the plant, and that's how it grows branches taller. So I'm not going to give you too many details on this, but these make up two distinct sections of the root. Up here, we call this area the zone of maturation, meaning it's the zone where the root is maturing and growing that vein. And this is where we start to see cells differentiating, meaning becoming different from one another. For example, the cells along the outside will start to become a protective layer. The cells in the center will start to become a vein. That's called differentiation. Now down here, these cells are just sort of stretching. All right, they're not differentiating, they're not mature, but we call this the zone of elongation. And it's the stretching of these cells that pushes the root crop through the soil. All right. And remember, there's a purpose to all of this. Let's zoom down just a little bit. The purpose to this root is to pull in water, NPK, sorry, I'm checking the clock, water, NPK, etc., in through that cell membrane into these cells. That water and NPK will travel through diffusion up here to the vein. And once it hits the vein, it can move a lot faster. And we'll learn more about that when we learn about vascular tissue like stems. But the point of all this is that the minerals and the water form a solution. So N, P, and K, which are the minerals, we would call those the solute. The solute is the thing that dissolves in the water. So if we're making Kool-Aid, the Kool-Aid powder would be the solute. So that N, P, and K, that solute, dissolves in water. That would be our solvent. That's the water in the Kool-Aid, or in this case, the water in the liquid inside the plant. Got that dissolving in the water to form a solution. A solution is a dissolved mixture of both some sort of liquid, maybe also gas, that's another story, we'll get there in chemistry, some sort of liquid with lots of things dissolved in it. All right, so form a solution that travels upward. And so that's the whole point of the root. Why does it travel upward? Well, you know the answer to this one. It travels upward because we're trying to get these nutrients and this water up to the leaves so that we can do photosynthesis. So this is what we went over the other day. We know that plants need sunlight, carbon dioxide, and water, and they produce oxygen as a waste product and glucose. All of this happens down here in the chloroplast. I don't have a ton that I want to add to this, but I do want to review it with you. So this is our chloroplast. It's a tiny little dot. You've already got this glued in, I believe. It's a tiny little dot, little organelle in the cell. It takes in light, which gets absorbed by a pigment called chlorophyll, and water. It uses those to produce an energy carrying molecule, ATP, which brings that energy from sunlight over to the Calvin cycle. Calvin cycle turns CO2 into glucose, and we recycle that energy carrying molecule, and we dump oxygen out as a waste product. That's what's going on here. There's not a lot I want to add to this. There's so much detail this, but you really don't need it until you get to college if you decide to study plants. But yeah, I'm just gonna hold back and move on. Switch my colors. Gotta preserve the color palette. You know what I mean? You can't just be throwing color palettes away. So I want to show you a little bit more about the physiology of the leaf that holds these very important organelles. So what we've got here, this top picture, this is a cross section of a leaf. So if we chopped a leaf in half and looked down the edge of it with a microscope, this is what we would see. Okay, and this was taken with a scanning electron microscope at SEM. You remember that maybe from the first unit. So there's a lot of layers going on here, and I want to talk through these with you. This top layer, up here at the very top, you can see it's kind of shiny. That top layer we would call the waxy cuticle. It's waterproof. It's what keeps these cells down here from being damaged, but it's also why water rolls off the top of a leaf instead of soaking into it, sort of like water sometimes soaks into your skin. So that is our waxy cuticle. Okay. Its literal whole job is to be waterproof. This also protects plants from bugs that want to eat them. It protects them from dust and debris. It's just like a really solid jacket. And then we see this little layer of empty cells right here. It's kind of sad looking, actually. It's just a bunch of dead cells. There's nothing going on here. These are actually the same as your skin cells. They're not serving like an active alive purpose. They're literally just there, again, for protection. So this is what we would call the upper epidermis, okay? Epidermis means skin, and this is the upper layer of skin on the leaf. Then we move into the exciting part of the leaf. All the stuff in the middle, we call the mesophyll. Meso means middle. So you see we've got these cells that are all sort of stacked upright right here. Okay, these are called the palisades. Palisade means like column, like in a palace. And we call them that because they look like a palace. And so that chunk right there, I'll label this palisade. And those absorb light. That's their job. And then right below them, we've got this sort of spongy area. You see how these look maybe a little bit more like a sponge? I don't know. Some people thought they looked like a sponge. I'm not arguing. So that area is called the spongy mesophyll. And that layer absorbs CO2. And together, these make up the mesophyll of the plant. Meso means middle, and phyll means leaf. I'm like 80% sure. It's been a while since I checked on that one. All right, as we move farther down the leaf, see a couple other things here. Here's the most important one. Do you see this little hole and this little hole? It kind of looks like a little mouth, like if we looked at our mouth from the side. Oh, why'd you do it this way? All right, we've got sort of the upper lip and the lower lip, and there's a hole in the center. That's what's going on here. We've got an upper lip and a lower lip, and that sort of hole in the center. Whoops, low battery. Hold on. Those are called stomata. We've got one here, one here, one here. I don't see a lot right here, and that's because this is protecting the center. But this is probably one over here. Yeah, let's label all three of these. These are stomata. If you only remember one thing from this drawing or this image, make it the stomata. Stomata are crucial because that is how CO2 gets into the leaf and how oxygen leaves the leaf. And we call that gas exchange. Because the stomata, they kind of take these deep breaths. They open and allow gases to pass through, and then they close and gases stop passing through. And that's how CO2 diffuses in and oxygen diffuses out. And then you'll see down here at the bottom, we've got another layer of these sort of dead looking cells. All right. That is our waxy cuticle, or I'm sorry, our lower epidermis. There isn't a tough waxy cuticle at the bottom. There doesn't really need to be. Not a lot of things land on the bottom of a leaf. They sort of land on the top. It's hard to have things land on the bottom. So there isn't really a waxy cuticle on the bottom. There is one more thing I want to add. See this center sort of circle right here? That's the vein. All right, that's where all the water is coming in to this leaf from. It travels through this vein like it's like a tube. I don't know that I can actually draw a line that you can see through that center. Let me try. Can you see that that's pointing to the center there? Yeah, kind of. All right, that's the vein. And you'll learn more about that soon on another page, but for now, that's the vein. Now, the point of the leaf is photosynthesis. So these mesophyll cells, that's where photosynthesis is happening. So we're going to pull these mesophyll cells, we're going to pull that idea down to here. Okay. These are pictures of plant cells. I'm going to label this again, mesophyll. Doesn't need to have a dash in the middle. I just keep running out of space. And so what we've got here is a light microscope. Actually, let's label this. This is an scanning electron microscope drawing at the top. And this is a light microscope. We're going to label that LM. SEM, LM. Okay, it's a nice little microscope review for you. And so let's take a look at this picture and see what we've got here. You see these sort of brick shapes, all right? That outside is the cell wall. We've seen this in other drawings. You can't really see the membrane, but it's there. So we're going to do and membrane. Membrane, that should be a B. Goodness gracious. Okay. And those are both made of cellulose. Cellulose, you'll learn more about this later, but it's basically millions and millions of sugar molecules stacked together. And that's what makes up wood. So wood is just bajillions of cell walls stacked together. And then inside, you see those little green dots? Those are the actual chloroplasts. The thing we were looking at right here. So let's point to one of those. I like this little one on the edge right there. That's our chloroplast. But there's obviously about 100 in here. Gosh, there's so many things I could teach you about chloroplasts. They're so cool. But alas, I've got to keep this appropriate for the first year of biology. So all you need to know at this point about chloroplasts is they are the organelle that photosynthesizes. All right. I'm guessing that you are just so intrigued you want to dive deeper into a chloroplast. Lucky for you, I've got one more micrograph here, one more image. All right, this final image, this is taken with a TEM, transmission electron microscope. So we're going to label that. Okay, I'm going to label it down here at the bottom. TEM, transmission electron microscope. If you're trying to remember what those are, you can turn back to Unit 1 and take a look. But here's what's happening here. This is our chloroplast. And it's sort of like a little lump. This would be the bottom of the cell, and this would be sort of pointing toward the sun. So light would come in like this. And these are those little pancake stacks that we talked about the other day. They're technically called grana. Each stack is called a grana. Individual stacks are called individual stacks are called a granum. Together they are the grana. But they're just stacks of those little thylakoid membranes, and they're filled with chlorophyll. Okay. So let's pull out a couple details I'd like you to see here. If you look closely, you can see that the chloroplast has this outer membrane and also an inner membrane. Okay. Which is unusual for an organelle. Which had people wondering for a long time, and we've got some ideas of why that might be. I'll tell you about those later in the unit, but that's what we call a double membrane. Okay. And photosynthesis is what happens in the center of this. So let's pull out a little thylakoid sac here, or a little stack. So we've got this stack of pancakes here. Okay. We call the whole stack a granum. And we call the individual pancakes in the stack thylakoids. So it's a granum of thylakoids, or a stack of pancakes. And so what's actually happening in this picture is photosynthesis. Photosynthesis happens in the thylakoids. Actually, I'm going to switch colors on this one. So photosynthesis, we got two words here. Photo and synthesis. What does photo mean? Think about it for a second. Well, photos capture light, all right? And that's what we see as an image. So photo means light, and synthesis means to make something. So what is photosynthesis making? Well, photosynthesis is literally light making carbohydrates. And so that's what's going on in this picture. Photosynthesis is light making carbohydrates, and the way that we're able to do this is because the thylakoids are filled with that pigment that can capture light, capture the energy of light, and that is, do you know the pigment? Chlorophyll. All right. Hang in there, folks. If you need to take a quick break, you can take a quick break. If you need to pause and take a look at your notes and sort of process them, you can do that. But I've got one more chunk of physiology to teach you today. I'm wondering if we're even going to get to. We might start homeostasis, but we might not finish it. Let's find the next thing. So we've got some things going on here. We've got water and nutrients traveling up from the roots. We've got CO2 being captured, CO2 and light being captured in the leaves, but they have to meet somehow. And the carbohydrates that are made up here, they have to get back down to the roots to feed the roots. So we need a way for things to travel through this plant. Luckily, we have, oops, just kidding, that's osmosis and tonicity. Hey, everybody turn to 186. Here we go. We'll read this another day, but for right now, I just want you to glue in this paper. This is a paper that's going to show us a little bit about the vascular system inside a plant. All right, so this, go ahead, pause the video, glue this to 186. And then when you're ready, I'm going to give you it will probably be a quick overview. I have so many exciting things to tell you about plants, but I can't tell you all of them. Ugh, heartbreak. I can't. No, okay, here we go. So we need some things to travel up from the roots, and we need other things to travel down from the leaves so that every part of the plant gets the sugars it needs to survive, and that the materials to make those sugars can travel to where they need to be. So we're going to go really light on this one. Plants have two major types of vascular tissue, xylem and phloem. Xylem and phloem. A couple different ways I've heard people try to remember these over the years, but basically, phloem allows things to flow down. It actually flows both directions, though. But xylem only allows things to flow up. So for xylem, we've got a one-way flow of water and minerals, but you know the name of the minerals at this point. They are nitrogen, phosphorus, and potassium, NPK. And xylem is basically a giant straw. There's no cell walls in between. It just like right up the stem. Phloem is a little different. This is a two-way flow. Things can flow up from the leaves this direction. They can flow back this direction. They can head down to the roots. And this is how water and food move around the cell. Obviously, you know better than to say food at this point. When we say food for plants, we mean glucose. Sugars, carbohydrates, same thing. And so if we were to cut this stem across the middle and look down into it, this is what we would see. So right here, when we say vascular, vascular means vein. So this right here, this is a vascular bundle. Doesn't that look kind of creepy? It always kind of freaked me out. So this is a bundle of veins. Oh, I should have used the darker color. Here we go. I'm going to go red on this one. Here we go. That's a vascular bundle. So let's label that. Basically a bundle of veins. And if we look really closely at this vascular bundle, we've got this sort of fibrous cap. This will eventually become the bark. Okay, so let's label that one. This big fibrous area. And that gets pushed outward as the vein grows and eventually makes this on the outside, which we call the bark. Which is just another word for epidermis, which is just another word for skin. But behind this fiber cap, we see two things. We see a bunch of little tiny cells, and we see a bunch of larger cells. The little tiny cells, those are our phloem. And things are traveling up and down them in both directions. These larger cells, they're kind of dead inside. It really is just a giant straw. And that is our xylem. And they're organized into these vascular bundles. Now, how they're actually organized depends on what kind of plant you're looking at. So let's down here a little bit. We will add some more notes here and here at some point, so try to leave those areas blank. But all flowering plants fall into one of two categories. So we call flowering plants angiosperms. We're going to add this at the very bottom. Okay, so if we were going back to our unit where we did a lot of taxonomy, angiosperms would be one big group. Okay. Flowering plants. Can you see that? Let's see. Oh gosh, that's awful. Hold on, I can do better. There we go. Flowering plants. And then that one big group is divided into two smaller groups. These are the ones you really think of. Most plants fall into this category up here. They have two little baby leaves that open up, those two cute little leaves at the beginning. They've got leaves with veins like this. They've got those vascular bundles that you saw up ahead. And their roots have a long center and then lots of things branching off of it. This is sort of our classic plant. This is called a dicot. Okay. However, some things like corn and lilies, they've got these leaf veins that go all parallel to one another. They've got vascular bundles that are much smaller, and they've got sort of fibrous roots. These are called monocots. And they're named after how many baby leaves they make. Those first two leaves are called cotyledons. If it makes two, it's a dicot. If it makes one, it's a monocot. So these are our two groups of angiosperms. All right. We're going to continue on 185 another day, but I do want to get us started on homeostasis. All right. So if you would turn back to 184, this is your homework. Some of you have started on this already, some of you haven't. But the idea of homeostasis is going to be really important to how these materials move from the roots to the leaves and from the leaves back to the roots. So this is all about cell transport. Osmosis is kind of like the how it happens. But there's sort of this larger process happening because everything in a plant needs to maintain that steady internal condition. It needs food, but not too much, water, but not too much. And the idea of that steady internal condition, that's homeostasis. So this is page 185. Would you please turn to page 185? Let's at least get started on the idea of homeostasis. Great. So this is going to be homeostasis and cell transport. Homeostasis and cell transport. Oops, we're going ran out of space. Homeostasis and cell transport. So homeostasis is the idea that cells and entire organisms like you and me must maintain steady internal conditions. You remember that from the first unit, I hope. Okay, and if you get a little stuck on that, you can go back to unit 1 and refresh your memory a little bit. But what does that mean for plants? Okay, well, plants need to be able to absorb nutrients to maintain those steady internal conditions. They need to balance the amount of water they have. Not too much, not too little. And they need to be able to get rid of their wastes, and that's true for all of us. Expel. Are there 2 L's on expel? I'm going with just the one. One must not waste. So expel waste. All of these things happen in the cell membrane. The cell membrane is what regulates these. So you've learned a lot about the cell membrane in your cell membrane homework, which is on page 180. If you haven't done this homework, oh my gosh, stop what you're doing, go do it. Pause this video and then come back. But if you have done this homework, go back, take just a quick look to refresh your memory on what's going on in the cell membrane. And once you're back here, let's talk about the 2 different ways materials can enter and exit the cell. We can sum these up in 2 words, either passive transport, meaning it doesn't require a lot of energy, or active transport, meaning it does require energy. So here we go. Passive transport, active transport. So in passive transport, it can happen on its own just based on the structure of the membrane and the materials involved. Energy is not required. Active transport, on the other hand, cannot happen just based on the structure and the materials. For these, energy is required to make it happen. So let's talk about an example of this. In fifth period yesterday, Jaden offered us a gummy bear, and we put that gummy bear in water. That was yesterday. Here's today. Let me put this little guy down and see if we can zoom in enough for you to see him. Oh my gosh. So yesterday it was about that tall. So he's doubled in height. And look, when I jiggle him, do you see that he's kind of made of jello at this point? So water was transported into this gummy bear. What do you think? Was it passive or active? Well, did we have to add any energy? This gummy bear's not alive, so he can't supply any energy. This is actually an example of passive transport. Passive transport works because materials are always moving from an area of high concentration to an area of low concentration. So let's draw what that looks like real quick. Drawing a box, drawing a double membrane through the middle. I'm labeling this side the outside, and I'm labeling this side the inside. Talking about the cell, or maybe we're talking about the inside of the gummy bear. So we have a bunch of water molecules outside the gummy bear. All right, let's draw our little water molecules. Little Mickey Mouses. Oh, they're so cute. Gummy bears, though, they're squishy, but they're not like sloshy. So we can assume there's very little water inside a gummy bear. So naturally, these materials are going to, the water is just going to diffuse from an area of high concentration to an area of low concentration. Could be water, could be something else. This is why we really hate it when people spray body spray in the bathrooms, because great, the concentration in the bathroom gets really high, but eventually it's going to diffuse outward and move from that high concentration in the bathroom to the lower concentration in the hallway, causing all of us to have an asthma attack. The word for that is that movement, that natural movement from high to low concentration. The word for that is diffuse. Diffuse from high to low concentration. What would you have to do at this point to get the water out of the gummy bear? Well, you could take it out of the water and it might dry out because now there's more water inside than in the empty space around it. That water might diffuse outward. But if you wanted to keep getting water out of the gummy bear, if you wanted to turn it into a little shrivelly gummy bear, you would have to somehow pump the water out of it. Maybe you could dry it out, turn it into steam in the oven. Because you're moving things against their concentration gradient at that point, you would have to add energy. All right, so and that's what's going on in active transport. So maybe inside the cell, we've got plenty of nutrients. Outside the cell, there are fewer. Just like inside you, there's more food you're currently digesting than there is just like in the space around you. So there's only a little bit here. I hope you can imagine that moving this food from an area of low concentration to an area of high concentration, it's not going to do that naturally. So we actually have to put some work into it. So active transport, this is why it requires energy, because you're moving materials from an area of low concentration to an area of high concentration, the opposite of the natural flow of things. And in that case, since it's not the natural flow, well, let's finish this up. Materials diffuse from low to high concentration. However, the word diffuse is no longer appropriate because diffuse means that sort of natural flow. And I wrote it, now I'm regretting it, and I'm apologizing. I'm not going to white it out. I'm going to cross it out. They're not diffusing. Those materials are moved. It requires energy. Okay, they're sort of moving against that concentration gradient, against that natural flow. This is why I wanted you to do your osmosis reading first, because that osmosis reading gives you a lot of words and a lot of background to understand what's going on with our gummy bear there. Now, another thing we could do is we could take a sample of plant cells, maybe like a little slice of potato or something, and we could put it in a bunch of different solutions, a bunch of different concentrations. Maybe one that's just water, maybe one that's water with a little bit of salt in it, maybe one that's water with tons of salt in it. And I think we would see a movement of water based on the amount of dissolved materials. Now, this is where things get a little bit trickier. What I just described is called osmosis. So if you haven't read this reading, you might want to take a look just at the top of this. Basically, water will move toward a higher solute concentration. Okay, if I've got a bunch of salt here and not a lot of salt here, we'll actually see water move until the concentration of solute is even on both sides because we've sort of watered it down on this side. That's the idea of osmosis. Osmosis is technically an example of passive transport. This is a natural movement of water because we've got so much stuff on this side. All right, and this is where I want to be careful because passive and active transport can get a little bit tricky. And we'll dig into those a little bit more another day. So I'm going to stop the video here. But if you haven't already done this homework, do this homework. I think it'll help you understand some examples of passive transport. And then you've got some practice problems on the inside to take a look at. If you haven't finished this, this is your homework due Tuesday. And then obviously finishing this video is also homework due Tuesday. Nice work today, y'all. Thank you for hanging with me. And Mr. Diaz or our substitute will have a couple extra directions for you. And if you've already finished your IOA, congratulations. And if you haven't, it's coming soon. You can practice. Whoops. I just took a picture. How did I do that? Okay, here we go. I want to stop share and I want to stop recording. Love you all.